Assalamualaikum and good afternoon. Good afternoon to Dr. Propose and Dr. Azman and Dr. John. I am Nur Kamilah binti Muhammad Nordin, and I am the student and the supervision of Dr. Azman and Dr. Ahmad Nizam. So today I am going to present on effect of plant growth regulators on the shoot biomass and antioxidant activity of Persicaria odorata. Okay. So for as the introduction, biomass refers, what is biomass? Biomass refers to the mass of living organisms, including plants, animals, and microorganisms, or from a biochemical perspective, cellulose, linear sugars, fats, and proteins. As for plant biomass is an important factor in the study of functional plant biology and growth analysis, and it is the basis for the calculation of net primary production and growth rate. Okay, so I would like to introduce a bit on the parts of the plant. So we could see here there are certain organs that are labeled there. So shoot tip, petiole, leaf, lateral shoot, lateral bud, stem. These plant organs can be cultured and introduced in vitro. Okay, so these are some of the examples. The culture from Pinelia ternate. So this one, okay, there should be a circle there. Okay, never mind. Okay, so what are these? What do you think are these? Okay, actually these are root cultures. Okay, these are root cultures. So how can we get this kind of vision? Is when PGR, plant growth regulators, supplemented in the MS media, which has been told by Dr. Azman. And this one is callus, but the first one is callus produced from petiole, and B is callus too, but produced from the leaf. Okay. Okay. So as for the literature review, okay, why is it black? Okay. Okay. So plant growth regulators that are most used are auxin and cytokinin. Auxin is required by most plant cells for division and reinsertion, and cytokinin for shape proliferation and shape morphogenesis. So these are the most used in experiment. So for auxin, we have IAA, NAA, 2,4-D, IBA, cytokinin, kantine, BAP, zetin, and isopentenyl adenine. Okay, so why Persicaria odorata? Okay, so Persicaria odorata, which is also known as kesum or Vietnamese coriander, from Paul Sky et al. The main compounds identified from the wild type are methyl gallate, catechin, Christine. Christine is mostly found in any articles regarding Persicaria odorata, and also by Rafida et al. It is high in antioxidant activity due to high amount of polyphenolic compounds. So these are the compounds found from the wild type of Persicaria odorata. So my objective is that I want to culture this and produce them in vitro. Why? Because you can control the temperature, you can control many variables, and you can produce expected outcome from the parameters that you control. Okay. So for the first one is to optimize the use of plant growth regulators in the micropropagation of Persicaria odorata, and the second one is to study the antioxidant activity in the shoot biomass. Okay, so this is the overview for the methodology. Okay, so this is the wild type of kesum. So in order to introduce kesum in vitro, surface sterilization needs to be done. So in this experiment, organ cultures used under the explant and shoot. So the incubation period before PGR can be introduced is 6 weeks and 16-hour photo period. Okay, so what is 16-hour photo period? 16-hour photo period is when 16 hours the culture is exposed to light and another 8 hours in dark condition. Okay, so okay, maybe I just have to. Okay, so for the PGR use is single BAP and the combination of BAP and NAA. Okay, so the fresh shoot biomass was also extracted in the ratio of sample to distilled water 1 to 50. So the analysis used here is DPPH and FRAP assay. Okay, so this is basically the surface sterilization made, and this one is how you introduce PGR in the MS media. MS media is the medium used to produce plants in vitro. Actually, there are so many types, but MS media is the most used one. Okay, so here comes the result. Okay, so the result here, so this is the effect of single PGR, which is used here is BAP. So the first row shows the result of control, means the media has no PGR at all. Okay, and for the treatment used is BAP, which is the concentration used 1, 2, 3, and 4. And from here, we could see that 2BAP produces highest fresh weight. Okay. However, the result is comparable to the control, which we could see that the control produces slightly higher weight. However, the number of shoot produced is still the highest. So we choose this concentration, 2BAP, to be combined with NAA. Okay, so the combination treatment used is the combination PGR used is BAP and NAA. So the first row shows the control explant, which the fresh weight produced is 127, and the only concentration change is NAA 0.05 up to 0.2, and you can see here the highest and the best result is produced by 2BAP and 0.2 NAA, which is 219, which is very significantly different in compared to the other treatments. Okay, so as for the shoot length, it produces 3.22 cm, and for the number of shoot per explant is 4.13 shoot per explant. Yeah. Okay. So these two slides show the explain on different. Okay. Okay, so this is the result for the antioxidant activity for the shoot biomass of Persicaria odorata. Okay, so here we could see that from FRAP assay, 2 milligram BAP, which is because we chose the best PGR that produces the highest shoot biomass. We could see that the value is higher than the control, which is very high, 150.09, sorry. And for so this indicates high antioxidant activity. So as for DPPH, it is lower than the control. So what does this indicate? Lower IC50 value indicates higher antioxidant activity. So high value of FRAP assay and lower IC50 value of DPPH that we want to achieve. Okay. Skip. Okay. So the combination of PGR helps in increasing the shoot biomass compared to single PGR because we could see the role of NAA just now. So high shoot biomass contributes in high content of secondary metabolites that act as antioxidant. Okay, secondary metabolites are compounds found in plants that also act as antioxidants. So it is concluded that high biomass of Persicaria odorata gives rise to antioxidant content. The relationship between high biomass of Persicaria odorata with good antioxidant activities may be further investigated, such as maybe in the synthesis of silicon nanoparticles.